Within the spectrum of human variation, the blue iris is a rare biological feature, appearing in roughly 10% of the global population. But despite how it looks, the human eye does not actually contain any blue, green, or hazel pigment. The color of an eye is determined by the concentration of a dark biopolymer called melanin within the iris stroma. In brown eyes, high concentrations of melanin act as a light sponge, absorbing incoming wavelengths and projecting a dark color back to the observer. A blue eye, however, is entirely devoid of melanin. When light strikes this transparent tissue, the long red and orange wavelengths pass straight through and are absorbed at the back of the eye. But the shorter blue wavelengths collide with the collagen fibers within the stroma and scatter outward. This structural reflection is known as the Tyndall effect. From a survival standpoint, this lack of pigment is a disadvantage. Melanin protects the retina from ultraviolet radiation. Without it, carriers are significantly more susceptible to macular degeneration and ocular cancers like uveal melanoma. Because blue eyes offer no physiological protection and increase the risk of disease, their survival contradicts the assumption that traits are only preserved if they offer a physical advantage. To find out why this trait survived, we have to look past anatomy and into the nucleus of the cell. The central driver of eye pigmentation is the OCA2 gene, which encodes the proteins necessary for producing melanin. In the ancestral state, DNA folds into a chromatin loop, bringing an enhancer to the OCA2 promoter to produce melanin. In the derived state below, a single-letter error in the HERC2 gene breaks this loop, causing a 5-fold reduction in OCA2 activity. In 2008, researcher Hans Eiberg analyzed blue-eyed individuals from Denmark, Turkey, and Jordan. He found that over 97% of them shared an identical, highly conserved sequence of genetic material stretching across half of the HERC2 gene. This identical genetic landscape points to a founder mutation. It suggests that this specific DNA error occurred precisely once, in the germline of a single prehistoric individual. This means that nearly every blue-eyed person on Earth today is carrying a direct, unbroken copy of that original mutant DNA strand. Paleogenomics traces this founder event to the Mesolithic era, roughly 14,000 to 17,000 years ago. DNA extracted from ancient skeletons, such as the Villabruna remains, revealed a startling physical combination. The original carriers of blue eyes were Western hunter-gatherers who possessed deeply dark African-like skin. This chronological separation proves that blue eyes evolved millennia before light skin. The trait was not a byproduct of global depigmentation or a generalized loss of melanin, but an independent genetic event. The mutation spread as these hunter-gatherers interbred with expanding populations of early farmers and nomadic steppe herders. Yet simple migration doesn't explain why a disadvantageous trait would reach such high frequencies. Statistical models shows that over the last several thousand years, the blue eye allele faced a selection coefficient of 3.6% per generation, one of the strongest signatures of positive selection ever observed in the human genome. Evolutionary theories suggest this was driven by sexual selection and paternity confidence. Because the trait is recessive, it acted as a visible marker of genetic inheritance. Men who preferred blue-eyed partners gained a reproductive edge by maximizing the certainty that they were investing their resources into their own biological offspring. While all blue-eyed people share a single ancestor, the massive population expansions of the last 14,000 years ensured a dilution of that ancestral DNA. Modern blue-eyed individuals are generally no more closely related to each other than any two random people from the same region. This genetic history also challenges the binary models of inheritance taught in many biology classrooms, which often suggest that two blue-eyed parents can never produce a brown-eyed child. Human pigmentation is actually a polygenic system. While HERC2 is the primary regulatory element, at least 16 different secondary genes interact to determine the final density of the stroma and the volume of melanin produced. This occurs through epistasis, where specific modifier genes can override the HERC2 mutation entirely, allowing for brown-eyed children to be born from two blue-eyed parents. This chart from the HI-RISPLEX-S system shows how that complexity impacts our ability to predict eye color. While we can predict the extremes of the spectrum, blue and brown, with over 93% accuracy, intermediate colors like green and hazel remain a difficult puzzle. We also see the resourcefulness of biology in isolated populations, like the Melanesians of the South Pacific. Their light pigmentation is caused by a mutation in a different gene entirely, proving that the human body can achieve similar results through completely different evolutionary pathways. Ultimately, eye color is not a boundary that divides us, but a molecular echo from a single hunter-gatherer living 14,000 years ago. It serves as a living reminder of the shared, interconnected history that defines our species.